In Chapter 9, we are going to talk about linked genes, or genes that are co-located on a single chromosome, and how we can use those linked genes to make genetic maps, or maps of the physical location of genes on a specific chromosome. So let's start by looking at the difference between linked and unlinked genes. So in Chapter 8, we looked at dihybrid crosses. Now for our class, we actually did this with Chapter 5, but it belongs in Chapter 8. And Chapter 8 says that alleles of genes located on different chromosomes will be inherited independently. This is the law of independent assortment. And this refers to two genes that are located on two different chromosomes. So here we have gene A located on the orange chromosome and gene B located on the blue chromosome. Now when gametes are formed, you can get any combination of that parent A and B alleles. And we looked at this when we did dihybrid crosses, or those 4x4 Punnett squares. But every chromosome actually has hundreds of genes located on it. Genes that are located together on a chromosome are called syntenic genes. Alleles of genes that are located on the same chromosome are not going to be inherited independently. They are said to be linked to one another. So on the top here, we have chromosomes that show linked genes, where A and B are found together. Now, because they are found on the same chromosome, they are going to be inherited together. So if you have the big A, big B allele on your chromosome, you are going to pass the big A, big B allele together on that particular chromosome. And if on your other chromosome from the other parent, you have little a, little b together, you're going to pass on those together. Whereas when they were unlinked, you could pass them off to your next generation in any combination of the two parental alleles. But linked genes are found together, and so we can use this to help us make genetic maps. So linked genes are going to be inherited together more frequently than the corresponding alleles would be on two separate chromosomes. But sometimes recombination, we talked about this in Chapter 6, during meiosis actually separates them. So remember that recombination is when two chromosomes exchange genetic information. Now they do this because they need to stay together during meiosis and move to the metaphase plate together. But the result of it is an exchange between those two inner chromosomes. And so although linked genes are inherited together more frequently, sometimes we get new combinations. So if this were to be separated into 4 gametes, we're going to end up with one gamete that is AB, one gamete that has actually come from an exchange of information. So this chromosome is going to produce a gamete, big A, little b. This one is going to be little a, big B, because we've exchanged this end part of the chromosome. And then our last one is little a, little b. And those outer chromosomes are not involved in this recombination. If these genes are located close together, relatively close together, there's going to be less chance of a recombination event occurring in between them. Because of that, we are going to get a smaller number of recombinant chromosomes. If they are farther apart, there's more room and a larger chance that there's going to be a recombination event in between them. And so you're going to get a higher number of recombinant chromosomes. So let's talk about how linked genes can be used to produce a genetic map. Now we do this based on the frequency of recombination that occurs between those two chromosomes. So before we get too far into this, I want to talk about two quick vocabulary words. And then we are going to look at what happens when we study unlinked genes before we get into actually studying linked genes. So a couple of terms here. The parental chromosomes are going to be made from gametes that have the same combination of alleles as the original parents. So these were the outer chromosomes that we saw in the last example, and they are going to produce gametes that have the same combination as the parents. So these are the parentals. These ones in the middle where the recombination occurs are going to be our recombinants. These are the individuals that are going to have new combinations. They're different from either parent. We actually saw this in a dihybrid cross, which is why we are going to start by going through unlinked genes. So let's look at unlinked genes and see what happens with our recombinants. So let's look at a cross between two homozygous parents. Now originally, or when we were doing this in chapter 8, we were using homozygous parents like this. Two homozygous dominant with two homozygous recessive. We're actually going to split this up a bit, or change it up a bit, and we're going to start with parents that are essentially opposites. So they're homozygous dominant for one gene and homozygous recessive for another gene. So our parental generation combinations are big A, little b, and little a, big B. Those are the possible gametes we can produce. The F1 generation, if we were to do a dihybrid cross, all of our F1 generation individuals would be heterozygous. Now this is the same dihybrid heterozygous individuals from our previous cross. We just started with different parentals. Now we are going to do a test cross. Remember, a test cross was a cross between a homozygous recessive individual and an individual with an unknown genotype. We're going to do a test cross here, except we know what the unknown genotype is. It's this F1 individual. So we're going to cross this F1 individual with a homozygous recessive. So this F1 individual is going to produce 4 possible gametes. Now we did this before. This was our foil. So 4 possible gametes here. And they are all going to combine with only one possible. This one can only produce this combination. So we are going to cross these individuals. Now when this happens, the phenotypes of your F2 are all going to occur in a 1:1:1:1 frequency. They are going to be the AB phenotype, the little a, big B phenotype, the big A, little b phenotype, and the little a, little b phenotype. They are going to occur in equal proportions. So this is what is happening with the chromosomes. We have big A chromosome on orange, little b chromosome on blue. This is our parental generation, crossed with a little a on orange, big B on blue. This produces the heterozygous individual, big A, big B, little b. Now, and this is to produce the F1, so that F1 individual is going to be test crossed to a homozygous recessive individual. When this happens, this picture here represents the chromosome combinations that we can get. We can get big A phenotype with little b. We can get little a with big B phenotype. We can get big A with big B phenotype. And we can get little a with little b phenotype. So when this happens, these chromosomes here are the parental phenotype combos. And these ones here are the recombinant phenotype combos. So if you look up here, this big A, little b was this parent up here. Little a, little b was this parent up here. These ones are the new combinations of gametes. I apologize for the technical difficulties. My tablet is having issues today. So if we were to do this with a drawing or a picture of some actual traits, you can see what is happening here. So with unlinked genes, the original homozygous parents, we are looking at two different traits for color and wing size in fruit flies. So our normal wild type is there on the left. This is the tan color that we saw during our fruit fly lab with the large wings. Our book denotes wild type as just a plus, but earlier in the semester, we were writing our wild type as the letter with a plus. So the color for the fly is going to be E plus meaning the tan or wild type color. AP for apterous is our denotation for the other wing type. So the normal wing is going to be denoted by AP plus. So you are free to write with the letters or with just the pluses, as long as you understand what's going on and as long as I can figure out that you know what's going on. I prefer to write it with the pluses because I get confused easily. Our mutant or our homozygous individual is going to be homozygous recessive for ebony, which is this gray color, and apterous wings, which are these small sort of defective wings. So we are going to cross the double mutant with a normal fly. The F1 generation produces heterozygous individuals. So we have E AP plus with E plus AP. So let me write it the correct way. This is our genotype for our F1 individuals. So now we are going to cross one of these F1 individuals with a homozygous recessive or mutant fly. When we do this, remember these are unlinked genes, we see a ratio of roughly 1 to 1 to 1 to 1. This means that the genes are unlinked and segregate independently. So if you are given a set of phenotypes, you could identify that these are unlinked genes because these phenotype combinations are occurring in equal frequency. Now we still have parentals. We have the wild type here and the ebony apterous. Those are the parentals, and these other two are the recombinants. But all of these phenotypes are occurring in equal proportion because the chance of getting any one of the genes from the chromosome that E is on is independent of the chance of getting any of the alleles from the chromosome that the other allele is on. So what happens when they are linked? So we're going to do the same type of cross. Before I move into the picture, I just want to draw what we're doing. So we are going to do, I'm just going to write, I'm going to use C and D here. So we are going to cross little c, big D, with little d, sorry, big C, little d. So the F1 is then going to be big C, big D, little d. We're going to cross this with our double mutant or double recessive, and we're going to look at the frequency of offspring. All right, so we have the same wild type male, normal body color, normal wing size, and this time we're going to look at a different color. Now this color, the black color, is located on the same chromosome as that apterous gene. So I know the color of these flies looks similar. We are actually looking at a gene for black color instead of ebony. Ebony is kind of a dark gray. Black is going to be darker than that. So we are going to cross our double mutant, the black apterous female, with a normal wild type male. F1 generation is going to be heterozygous. You can see that right here. And then we are going to test cross this female with a homozygous, so this female here, or this type of female. I'm sorry, that arrow's in the wrong place. The arrow should be here. We're going to test cross this F1 female with this homozygous recessive male. Now, if you look at the phenotype information that we get from this cross, the 4 different phenotypes do not occur with equal frequency. We see that these phenotypes that were parental, the wild type and the black with apterous, occur far more frequently than the recombinants, the black body with normal wings or the normal body with apterous wings. Now this suggests, because it occurs much less frequently, that the A, P, and the B gene, the black and apterous genes, are linked together because those traits don't separate very frequently. We can actually calculate it. The percent of parents is 93%. The percent of recombinants is 7%. So you can see that these occur far more regularly. They do not segregate independently, and so the genes must be linked. Those recombinants occur due to chromosomes crossing over. So there are several different possibilities for a crossover event when you look at these 2 genes. The first possibility is that the crossover event occurs on the other side of the centromere. If that happens, there's no recombinants for those 2 genes. The second possibility is that the crossover event can occur between the centromere and the 2 genes that you're talking about. If that happens, there's no crossover event and you don't get any new combinations of parental chromosomes. So in either of those situations, you only get parental gametes. The last one shown here is actually sort of the opposite of the second one. This is when the recombination occurs after your 2 genes. Again, you only get the parental combination of those gametes. But sometimes you get a crossover event between the 2 genes, and this is where you get your parental versus your recombinant gametes. Remember, those outside ones are not involved in the crossover, so they maintain the parental combination, whereas in the middle, we get the recombinant chromosome, or the new combination of alleles. So this diagram here shows the black and apterous crossing over. So if we have a crossover, or we'll look at the chromosomes first, these blue and orange are the parental chromosomes. The parental are going to produce the parental gametes. So no crossover occurs. We have the black and apterous together. On the outside chromosome, no recombination occurs. So we get the wild type. If we have a recombination event in between, we get the new recombinants. We get black with normal wings, and we get normal color with apterous wings. If the crossover event occurs outside of these genes, no crossover occurs in between, and we get only parental recombination. Parental gametes.